All right, now we're going to dive into cell energetics. So this is a recap of everything, kind of big picture from Unit 3. So the first part of this particular unit is to understand enzymes and how they contribute to cell energetics. So enzymes are proteins and also RNA too, but mainly they are proteins, and their job is to facilitate chemical reactions. So what they generally do is they make the reaction go faster, but they themselves don't get used up. So they just help things along, and then they go on and do it again and again and again and again. They do this by reducing activation energy. So a lot of the chemical reactions that take place in us require a certain amount of energy to make it happen. So enzymes basically help things along by reducing the amount of energy that it takes to make it happen. In terms of making the reaction go forward. They don't actually change the free energy that's overall released or required. They just kind of lower the activation energy. I'll show you that in a little bit. Most reactions in the body aren't fast enough to actually keep you alive, so enzymes are needed for almost all biological reactions. They're super specific. So think about the different types of models that we learned, kind of the induced fit model, where there are different enzymes that will catalyze very specific reactions, which is why enzymes are usually named after the specific reaction that they do. So there's, catalase. Now I can't think of any off the top of my head, but you get my point. I'll probably remember more as we go on. And they control the reactions of life. So here's an example of kind of how that works. So there's a substrate, which is the thing that the enzyme is going to act on. In this case, the goal would be to take this sucrose molecule, where sucrose is a glucose and a fructose stuck together. So just to break it. So the enzyme is going to come along, and you can see that there's this active site on the enzyme that acts, as kind of an induced fit lock and key type system that will bind to and create an enzyme substrate complex. So they're going to be together for a while. And then water comes in and actually does the splitting. So hydrolysis, if we remember, hydrolysis means to break with water. And then voila, we have glucose and fructose. They get released from the enzyme, and now the enzyme itself can go off and do the same thing again to another sucrose molecule. So this would be an example where the enzyme would be called sucrase. Enzymes usually end in ase. Okay. They are, again, very specific. They are not consumed, and they are affected by cellular conditions. So if you start messing around with the environment that the enzymes are living in, they don't quite work as well anymore because they're proteins. And if you remember, if you heat a protein up too high, it will denature. So temperature can affect it. Same thing with pH and salt concentration. We did learn about two ways in which an enzyme and a substrate can come together. So there was always that traditional model of lock and key, which is kind of very, very rigid and simplistic, where it was an exact fit. And then when they came together, hydrogen bonds would be formed, kind of fitting that key that is the substrate into the lock that is the enzyme. A little bit more accurate is kind of the induced fit model, where it's pretty close, but it kind of really locks in when the substrate forms the complex with the enzyme. And then that changes the shape a little bit, and then that's how you get the tighter fit. So the substrate kind of creates a conformational change that makes it all lock together. Enzymes, again, work by lowering activation energy, and they can speed up reactions. So if the job is to synthesize something or to, attach things together and make more complex molecules, oftentimes they just kind of orient things in the right place to make it a little bit easier. They would bring things closer together to join them. If the goal is to digest, then maybe it will conformationally change to induce some stress that will make it easier to break. I mentioned a couple of these. Temperature, pH, and salt can all affect an enzyme's ability to do its job. But there's others as well. There are activators, there are inhibitors, which, as you can imagine, will turn them on or off. And then the amount of enzyme available and the amount of substrate available also can impact function. So enzyme concentration, in general, the more enzyme you have, the more you can increase reactions because the likelihood of an enzyme coming in contact with a substrate goes up. But there's a limit. There's kind of a saturation point, if you will. So you can add more and more and more enzyme, but then, once you have an enzyme for every single substrate, adding more enzyme isn't going to make it go any more. The substrate kind of then becomes the limiting factor here. And so then you see this kind of leveling off at really high concentrations. The same is really true for substrate. If you increase substrate, you're increasing the likelihood that it would collide with an enzyme. But at a certain saturation point, all of the enzymes are occupied. So adding more substrate isn't going to have any effect. So it works to a point and then it levels off. Temperature. Every enzyme kind of has a sweet spot temperature of maximum efficiency. And if you get too high or too low, it's a bit like a bell curve. It stops working. Most human enzymes are going to work obviously around body temperature because that just makes sense. And when you go outside of that, it has to do with the particles moving, eventually becoming denatured, and so on. So here's an example. If this is an enzyme working in a thermal vent bacteria, well, then it probably has a higher optimum temperature because those bacteria are surviving in hot springs around 70 degrees Celsius. pH is the same principle. There's kind of a sweet spot pH for a lot of enzymes. Depending on where it is in our body, for example, it might be a different optimum. Our intestines are around 8, so it makes sense the enzymes there like it at 8, whereas ones in our stomach might like it around 2 to 3. And when you are modifying pH, you're really changing the amount of hydrogen ions available. So that messes with the 3D shape of the protein. And that's really what's impacting its ability to function. Salt concentration, just understand it's the same principle. It has to do with adding or removing charges and again, disrupting the shape. Think about the Dead Sea based on the salt concentration. Most enzymes are intolerant of that super extreme salt concentration, which is why nothing is alive in the Dead Sea. There are activators that can kind of make more magic happen. So they could be cofactors or coenzymes. Just generally know that these are things that can bind to whether it's inside the enzyme or temporarily or permanently, that can enhance its function. On the flip side, there are also inhibitors that can block a substrate's ability to bind to an enzyme and work. So it could be competitive. I don't know if I want, yes. Meaning that it's literally binding and blocking the active site and prevents the substrate from coming in at all to have a reaction take place. You can kind of get around this if you increase the substrate concentration because then just there's way more substrate than inhibitor, so the likelihood is that it's going to outcompete the inhibitor. It could be non-competitive, meaning it does bind to the enzyme, just not in the active site. So it's some other what we'll call allosteric spot, and it causes a change enough in the enzyme that it will no longer recognize the substrate. It's possible that it's irreversible. There are some toxins out there that will stick either to the active site or to an allosteric site forever and won't ever release and therefore kind of render the enzyme permanently unable to work. This picture, if you want to pause, just kind of talks a little bit about how that conformational change, whether it's in the active site or not, how that works. Chemical reactions are organized in a pathway. So this is kind of one of the big picture items of AP biology, just understanding that a lot of our processes have multiple steps with the goal that you're really increasing regulation. If it's just one singular step from point A to point G, then if that something happens that one step, all is lost. And it's kind of an all or nothing situation. It's either on or it's off. If you have all these fine-tuning steps between A and G, then you can kind of maybe overcome things if one of the steps becomes damaged or mutated in some way, but also it helps you fine-tune regulation. You can really get an exact product amount made by increasing or decreasing 6 enzymes versus 1. This also lends to the idea of feedback inhibition, where the product of a specific pathway, just by its increase in presence, can act as a physical inhibitor to one of the enzymes upstream. So in this case, isoleucine can act as an allosteric inhibitor on enzyme 1. So it will bind to enzyme 1 and prevent the pathway from continuing. Then as isoleucine decreases, well, then it's not going to be able to inhibit anymore, and then you can go through the whole pathway. So it's a way to control the amount of products that you're making. When it's in high supply, it shuts down the pathway. When it's in low supply, it allows the pathway to continue. Okay, so now we're going to break down into metabolism. And this is going to include both cellular respiration and photosynthesis. I just kind of talked about how pathways result from multiple reactions and multiple enzymes. Metabolism as a word just means all of an organism's chemical reactions, whether it's building things or breaking them down. Catabolic pathways are the ones that are breaking something down. Therefore, by breaking things down, you are harvesting or releasing the energy and then harvesting it. So your digestive enzymes are breaking down glucose. That releases energy that then we can store and use for other things. Anabolic is where you build things. So you need to actually use some of that energy or put in energy to build a structure from a simpler one. So thinking about how amino acids can form muscle protein. Kind of taking a chemical digression, there are 2 different types of energy that can be related to biology. So kinetic energy is the energy of motion. For biological purposes, that's mostly going to be in the form of heat or thermal energy. So heat is generated when atoms and molecules are moving all around. There's also potential energy, which is that stored as a result of position or structure. In a biological sense, potential energy is mostly to do with structure. A lot of our, fat molecules and other complex structures inside of us have energy in the bonds that they have created. So there's a lot of potential to release energy if those are broken. Remember, thermodynamics, energy cannot be created or destroyed. It's just kind of converted from one to the other. So we might break chemical bonds in order to be able to, move our bodies, fire our neurons, those kinds of things. Speaking of thermodynamics, it's just the study of energy transfer. That's what thermodynamics is. Now, if you have a closed system, this is where it's isolated from the surroundings. So I think of a liquid in a thermos. It's not really exposed or interacting with its environment outside of the thermos. This is not really true for any living organism. We are open systems. We regularly interact with our surroundings, and so energy and matter can be transferred in and out of us and our surroundings. Thinking about the different laws of thermodynamics, the first one is energy is constant. It can't be created or destroyed. It can only be transferred. So as I mentioned, a lot of times we could take the chemical energy of, let's say, a bear eating a fish and transfer that into mechanical energy or heat energy that the bear might give off. The second law of thermodynamics has to do with randomness or entropy, as it's called. Every energy transfer increases disorder. Just think of, as we're moving things around, your room gets messier and messier and messier, and it doesn't really take much. It just happens. Every time you do something, it can get messier and messier, unless you're putting in energy to make it more ordered. but energy is meant to increase entropy. So in terms of biology, a lot of times this has to do with us giving off carbon dioxide into the air and water and heat when we do cellular respiration. There is an equation that goes along with whether or not energy is needed or given off, and that is called free energy. It kind of looks at a reaction that's going to take place and determine where the energy is going. The change is called delta G, and reactions can either happen spontaneously, meaning that it does not require energy. Those are called exergonic reactions. They are giving off energy and they happen by themselves. Alternatively, there are endergonic reactions where energy is required. So in this case, you actually have to put energy in to make it happen. And therefore, by putting in energy, you have a positive delta G instead of a negative delta G. So here's kind of what an exergonic reaction looks like. Again, we're giving off energy. So whatever energy the reactants had stored in them is now less in the products. So you release a bunch of energy and now the products themselves within them contain less. And that difference is whatever the negative delta G value would be. Again, these are the spontaneous ones. On the flip side, endergonic ones are basically inputting energy into the reaction so that the products themselves are holding on to that energy. Therefore, you have to put some in to make it happen, and this would result in a positive delta G. So it's important to know that living cells are never at equilibrium. It never stops. There's always in and out. And the cells themselves do kind of a big picture thing called energy coupling. So our body has a really awesome way of combining and coupling reactions that give off energy with other reactions that require energy. So that way it's coupled in the sense that when one is giving off energy, then you have exactly whatever energy you would need to then drive the next reaction to ensure that both of them can happen. In terms of a reaction, this is how enzymes actually contribute to them. So the enzyme will lower the amount of energy that is necessary to make the reaction take place. So it doesn't change the amount of available energy in the beginning or the end. It just kind of lowers the hurdle to overcome in order to make that happen. And this is for endergonic reactions because, again, exergonic ones are spontaneous. So they don't need to have a lowered activation energy. In terms of energy that we're talking about, ATP is the primary currency. It stands for adenosine triphosphate, but it's the main source of energy that is used in coupling. So when energy is released in a specific reaction, it will be in the form of ATP, and then when it is used up, it is in the form of ATP that is needed to pay, so to speak. And the energy itself is arbored in the phosphate groups. So as you add them or cut them off, that's kind of where the potential energy lives. So when ATP, it got a little cut off there, is hydrolyzed, meaning that it is cut with water, you're basically chopping one of those phosphates off, and instead of being a triphosphate, it is now a diphosphate, or ADP. That phosphate is now available to do, or that energy that was used to do that can now go off and fuel other reactions. And then it's possible that other energy from the breakdown of other reactions can be used to regenerate ATP. It's important to know that ATP is recycled. It gets used and converted back and forth between ADP and ATP depending on what it's doing in a reaction. Things that it can do is it can help do active transport when energy is needed. It can also do other things like mechanical work, thinking about how our muscles contract. So now that we know a little bit about enzymes and how chemical reactions work, we are now going to feed into the idea of photosynthesis and cellular respiration. So, photosynthesis. comes from autotrophs, those organisms that are able to make their own food. And together with photosynthesis, they are called photoautotrophs. They make their own food with light. We, however, as humans, are heterotrophs. We need to actually eat other organisms to survive. So photosynthesis is unique to photosynthetic organisms. The magical place with which this happens is the chloroplast. So we learned about these prior in cell organelles, but they are basically the source with which light energy is converted into chemical energy, thinking back to thermodynamics, in the form of food. So most of this takes place in the middle portion of a leaf. So leaves are 3D. I know they're thin, but think of them as actual 3D structures. So in that middle portion, in the mesophyll as it's called, there's tons of chloroplasts. And within those chloroplasts, you can see kind of like rolls of quarters, thylakoids stacked on top of each other. They have spaces inside of them, kind of like the filling of an Oreo. And then there's stroma, which is the liquid surrounding it. So those chloroplasts really live inside the thylakoid. On leaves also, there are something called stroma, or stromata is singular. They are pores in the leaf. So that is where the gas exchange is taking place. So oftentimes when you look on the underside of a leaf, there are these pores that will open or close to allow carbon dioxide to go in and oxygen to leave. Chlorophyll, again, is the pigment, and a pigment is the actual component that's absorbing light. Here's the overall reaction for photosynthesis. So thinking about things that are needed to make a plant grow, they need water, they need light, and in this case, they need carbon dioxide. And that's going to be used to make C6H12O6, which is glucose, and they also give off oxygen, which is good because we need it to breathe. In terms of the chemistry behind it, basically water is split, and the energy that is released in the form of electrons is transferred with carbon dioxide, and together those things can make sugar, or glucose in this case. Photosynthesis together is two major parts. There are the light reactions, which is where the photo part comes in, and then there's the Kelvin cycle, which is actually the synthesis part. So the light reactions are meant to harvest the light and put it into some transport energy molecules that then are used in the Kelvin cycle to actually do the synthesis part of the sugar. And you can kind of see the ins and outs. Water is used to split, light is used to split water in the light reaction. So that part comes in first. And that's where oxygen is released. And then later is when the carbon dioxide comes in. Pigments. Pigments are structures that absorb specific wavelengths of light. So chlorophyll is a pigment that generally absorbs all the colors except for green, which is why most plants are green, and we observe chlorophyll as green. And there's two types. They kind of absorb slightly different colors within the visible spectrum around green. There's also other pigments, though. There's xanthophylls, which are reflective of yellow and absorb other wavelengths. And there's also carotenoids, which absorb pretty much the wavelengths that are not orange. So in terms of what color something is, the pigments are absorbing the colors that we can't see. The color that we can see is the reflected color that's not absorbed by the pigment. So you can kind of see that here in this picture. So the light reactions. Electrons in chlorophyll are excited. So within the chlorophyll pigment itself, there are electrons that can be excited by light. So what happens here is that they get super, super excited, and when an electron gets excited, it has a lot of energy. And then when it relaxes or calms back down, it can give off heat and other energy components when it tries to go back down to that normal state. Which is why we can talk about photosystems. This is kind of the overall center where it's happening. And it's kind of a complex where those excitable electrons live, but it also has some protein and multiple pigments involved. So here's kind of what a photosystem looks like. It's inside the thylakoid space, or in those inner membranes of the thylakoid. And so there's a ton of pigment molecules that are able to really absorb the energy from light, and they bounce their electrons and transfer energy to some special chlorophyll molecules. And once those are charged up enough, they shoot up to higher energy levels and can then transfer that energy to electron acceptors. Electrons can flow linearly, which is kind of the overall photosynthesis process that we're used to studying. They can also flow cyclically. So we're going to focus on the linear portion because that's really what is happening in terms of the photosynthesis that we talk about. Chlorophyll gets excited by light, which is always the very first thing. And then there are two major photosystems in photosynthesis for I guess, order with which they were discovered. Photosystem 2 comes before photosystem 1. So this magic happens in photosystem 2 first, and then that excited electron gets captured by a primary electron acceptor. And basically, there's a special transfer, and in order to replace this captured electron, water is split. And in the process of losing those electrons, then water is converted into oxygen. So that's where the oxygen component comes from, the splitting of water to kind of donate in response to the electron acceptors' electrons. Then those electrons get passed to photosystem 1, which comes second, via an electron transport chain. So those kind of get pumped and bounce down small energy levels, and in the process, ATP can be made by something called photophosphorylation. So phosphates are being added to ADP molecules to produce ATP molecules. That's where it's called phosphorylation. You're adding phosphates to generate energy. And this is fueled by those hydrogen molecules moving across a membrane. The energy carrier is NADPH. There's a P in there for photosynthesis. That's how I remember it. And basically it's carrying that hydrogen that can be used later. So big picture, photosystem 2 and 1 for the light reactions is to use solar energy to make some ATP and also some NADPH that will fuel the second part of photosynthesis, the Calvin cycle. So here's kind of the photo version of what that looks like. As I mentioned, there is a cyclical flow that's possible where really only photosystem 1 is taking place, and really it's just to refuel some ATP. So there's no oxygen made and there's no NADPH produced. It's not very energy efficient. It's mostly just to recreate some of the ATP that's needed later. And this is kind of showing you the idea of the photophosphorylation that's going to take place. It's important to know that both the mitochondria and the chloroplast have this chemiosmosis process to make ATP. So they both have this ATP synthase that utilizes a proton gradient. So the electron transport chain creates this proton gradient where there's a ton of hydrogens on one side and not a lot on the other. And as they fall through to kind of equalize, the ATP synthase is able to tack on phosphates to ADP and make ATP. So this happens in the second portion of photosynthesis, but it also happens in the third part of cellular respiration in the mitochondria. So this, as you can see, requires a membrane because you have to be able to pump things across the membrane. For the chloroplast, it's the thylakoid membrane, and for the mitochondria, it's the inner membrane. So really, how does this magic happen? The protons have come from water. So if you remember, we split water. So those H's are maintained while the oxygen is released. And then as things move down the electron transport chain and move along, those hydrogens are pumped and create a gradient. And then additionally, there are other ones that are kind of used and removed from the outside. So in this case here, you can see that from the outside spot to reduce NADP to NADH so that it can carry those protons, you're basically removing them from the environment. So pumping some into the interior and moving some from the environment create that proton force that then that differentiation is big enough to drive ATP synthesis. So that ATP that is made here, as well as the NADPH, can go to the Calvin cycle to create sugar from carbon dioxide. So the sugar, again, is going to be C6H12O6. So it's going to use those to generate some 3 carbon molecules that then can be used to make glucose. This is done in 3 parts. So first, we have carbon fixation, then we have reduction, and then we can regenerate Rubisco, or RUBP, which I'll talk about in a second. All right, so the first thing that happens, and I don't think you're really going to have to know the specifics of all of this, just kind of in general. So we have carbon dioxide, and we're going to bring in 3 of them. And this is going to happen twice because we're going to need to create C6H12O6, and you utilize 6 carbon dioxide molecules. So this is kind of happening twice. So we're fixing the carbon, meaning that we are bringing it in using a Rubisco molecule. And then we have to use up some of the ATP to kind of stick them all together. And in the process, we also are using some NADPH to make all of this stick together. So that's where the kind of the energy cost part of this is coming in. And then all of that magic happens and we get a 3 carbon sugar at the end. And this happens twice so that we end up with 2 of them. And then we have to use a little bit of ATP to kind of regenerate that Rubisco molecule. So it is recycled over time and can then take place to then carbon fix the next CO2 molecules and the next ones. So overall, importance for photosynthesis. 1, it's the source of food for the plant. So plants, please know, they do photosynthesis, but they also do cellular respiration. So they need to make their food and then they utilize their food. They also use it to make things like cellulose, which are a structural component of plants. Why do we also need photosynthesis as humans? Well, we eat them for food because they make food and then we can eat them, but they also produce oxygen with which we breathe. Okay, so on the right, you can see some of the things that we talked about for photosynthesis. It occurs in plants. It can also occur in photosynthetic bacteria and other things. You need carbon dioxide, water, and sunlight. It makes glucose. It gives off oxygen. In the process, it does generate ATP and NADPH that kind of is utilized in the Calvin cycle to make that glucose. This happens in the chloroplast, specifically in the thylakoid membrane, because that is where the proton gradient is made, and in the stroma. Plants then do photorespiration, so they do utilize the glucose that they made, and there was that proton gradient. As we transition to respiration, which is where we break down the glucose that has been made for food, or either that we made ourselves or that were consumed from other organisms, it requires oxygen. And in the process, we create carbon dioxide and water. We generate ATP, which is kind of the goal of breaking down food. And we have something called NADH. So this is the not photosynthesis one, which is an electron carrier or an energy carrier. This occurs in the mitochondria, in the membrane in the matrix, which I will talk about later. And there is something called oxidative phosphorylation. So this is similar to photosynthesis, where there is that gradient and you are using the gradient across a membrane to fuel phosphorylation and create ATP. All right, this brings us to respiration. Okay, so cellular respiration. This is going to include the catabolic reactions. So photosynthesis is really anabolic, so the goal was to build molecules and store energy, like in glucose. Catabolic reactions, if we remember, are ones that break things down to hopefully extract energy. So by definition, cellular respiration is to break down glucose or larger molecules into smaller ones. And again, it kind of got cut off there. So it's catabolic in nature. In open systems like us, we require energy to perform work. So we have chemical reactions that need to take place. We need to transport things around us. We have mechanical things that we need to do, like moving our muscles. So kind of in terms of energy flow, remembering that energy is not created or destroyed, we take in light energy, photosynthesis happens, we do cellular respiration to get energy, some of that's given off as heat, and so on. If you're looking at the specific equations, photosynthesis and respiration are opposites of each other in nature. Photosynthesis is endergonic because you have to actually put in the energy of light to make it happen. It's not a spontaneous thing. So if a plant is in the dark 100% of the time, no photosynthesis is taking place because you need light to fuel the reaction. Respiration, however, is exergonic. It is a spontaneous process that occurs, and it does not require energy. It releases energy and also produces heat. So in terms of the energy harvest, energy is released as electrons. They get super, super excited, go to high energy levels, and then kind of as they fall back down to their, normal, happy, unenergetic self with oxygen, which is known as the final electron acceptor, they give off energy during that process. There are specific intermediates that kind of help in this to prevent giant explosions of energy. So the idea is to kind of cushion the fall, make it happen in a couple of steps. So there's not just super excited electrons that fall down and have this explosion of energy that don't allow us to capture it all. So we do it in steps so that the body can actually harvest as much energy as possible. So enter NAD. NAD is an electron acceptor. It acts as like a little pickup or energy carrier. And when it picks up protons, or when it does this, it picks up protons and electrons. Remember, we need to kind of keep the charges balanced. So to do this, it creates NADH. And when it has NADH, that means it's carrying some electrons. And it shuttles them to the electron transport chain where it kind of drops them off at that last step. So it can drop off electrons and it drops off protons for that proton gradient that's used later. So here's kind of just the dynamics of how that works. NAD picks up in the process 2 electrons and 2 protons. So therefore it becomes NADH. And once it drops it off, again, it's a recyclable molecule, so it will turn into NAD once again. In terms of the electron transport chain, as I mentioned, oftentimes the electrons get really, really, really excited. And when that happens, some of those protons and the oxygen that is necessary come together to make water. There might be an explosive release of energy, which we don't really want. So to make that happen, we have the electron transport chain where things kind of bounce down in the process and create molecules of ATP as that happens. You still generate water at the end, and then oxygen is still the final electron acceptor. It just happens in a more structured way. So cellular respiration happens in 3 steps. Remember, photosynthesis happened in 2. This one is 3. There's glycolysis, and then there's the group of reactions known as the Krebs cycle, which kind of has a pre-step and then the cycle itself. And then the 3rd step is the electron transport chain and chemiosmosis. And here's roughly where they take place. So glycolysis occurs in the cytosol. No mitochondria necessary. So all organisms, because it kind of just takes place in the cytosol, can do glycolysis. and that's the initial breakdown step. There's a little bit of ATP generated, which I'll talk about in a little bit. Then these products of this glycolysis reaction called pyruvate can make their way into the mitochondria. And then there's this kind of oxidation step that converts it into acetyl-CoA, which is a molecule that it can then take place in the citric acid cycle. Please note, citric acid cycle and the Krebs cycle are the same exact thing. So a little bit of energy is made here too. But most of the energy is going to be packaged up into NADH, and then there's a second one called FADH2. These again are two electron carriers that basically carry the electrons to the third step, which is where something called oxidative phosphorylation takes place. And that's where most of the ATP is made. So glycolysis, first step. It literally means sugar splitting. So it's thought to be ancient because, again, all organisms can do it. It breaks glucose down into 2,3-carbon pyruvates. Now, there is a little bit of an investment. You need to actually put in a tiny bit of energy to make this part happen because hydrolysis does not always occur by itself and spontaneously. So you actually have to put into ATP. It makes 4. So when it says net gain, you get a total of 2. You had to spend 2 to get 4. And then you make a couple NADH molecules. In addition, we also make water. You do not need oxygen for this portion. So that's why all organisms can really do this, even the early prokaryotes, when oxygen wasn't available in the atmosphere at the time. So it doesn't create a lot of energy, and it's not really preferred if oxygen is available, but it doesn't need it. As I mentioned, there is the investment phase. And then at the end, you get a net production of 2. And what is occurring here is what we call substrate-level phosphorylation. It's basically just shoving a phosphate onto an ATP molecule. It's taking it off one thing and putting it onto ADP. So there is some kind of substrate that happens to have a phosphate on it, and an enzyme comes along and takes it off the substrate and shoves it onto ADP, turning it into ATP. So there's a specific molecule out there carrying a phosphate that will then make its way to create ATP. That's what's called substrate-level phosphorylation. And this occurs in glycolysis. We will also see it again in the Krebs cycle. So here's kind of the overall summary of what is occurring in glycolysis. So now we go to the second step, which is kind of the combination of oxidizing pyruvate and the Krebs cycle, or citric acid cycle as it's known. So here's what the mitochondria looks like. This particular portion, the second portion, happens in the matrix, which is the fluidy part in the middle. And then transport chain requires a proton gradient, so we're going to have the membrane there, and this takes place in the inner membrane. So all that happens in the intermediate step is that this is where carbon dioxide is given off. So the pyruvate molecules that come in from glycolysis are converted into something called acetyl-CoA. And in the process, you are now able to give off carbon dioxide because you lost a carbon, and you also produce some NADH. Then the acetyl-CoA molecule is what proceeds on to the Krebs cycle. In this process of the Krebs cycle, you get a couple of things. You gain a couple more ATP, you gain some more NADH, some FADH2, which is a second type of electron carrier, and then noting that the ATP produced here, similar to glycolysis, is done via substrate-level phosphorylation. Here's what it looks like. You would never have to know the intricate steps of it, just kind of know what is occurring on. You can see down at the bottom, that is where the ATP substrate level phosphorylation is taking place, and you can the net gain of different things. All right, so to kind of summarize what's going in and what's going out, brings us to the third step, oxidative phosphorylation. So oxidative phosphorylation has to do with the oxidation occurring. There's not any one substrate that's just carrying a phosphate. It has to do with the proton gradient across a membrane. So really this portion is very, very similar to photosynthesis and what's occurring there. So in terms of the electron transport chain, electron transport is occurring in the inner membrane. At the end of all of this, about 26 to 28 ATP will be made by the process of chemiosmosis. So chemiosmosis, excuse me, is really the process of the proton gradient being pumped across the membrane, and that then diffusion through ATP synthase fuels the production of ATP. So I'll show you what that kind of looks like. In terms of the electron transport chain, NADH, which was one of those carriers, drops off its electrons, and then the hydrogens will go towards the proton gradient. FADH2 is also an electron carrier, but it can't hold quite as much. So it drops the electrons off at a little bit lower energy level. The hydrogens will also go towards the proton gradient. So these things will go slowly down the electron transport chain to the final electron acceptor, that is oxygen. And so some of those hydrogens will combine with oxygen, again, to facilitate the proton gradient to make water. This does not directly make ATP, so there's no substrate level phosphorylation occurring. It's going to be really due to the chemiosmosis part in a second. So here's kind of how that proton gradient is created. In the process of NADH and FADH2 dropping off their molecules, hydrogen go into the intermembrane space, and then some are removed from the matrix in the formation of water. So that creates that huge gradient that can then be utilized by the ATP synthase. So it allows hydrogens to kind of move their way back through, and in the process, they generate ATP. So overall, when all is said and done, you get 2 ATP from glycolysis, 2 from the Krebs cycle, and then somewhere around 26 to 28 from the third step, the oxidative phosphorylation or electron transport chain. When you add it all together, then it ends up being about 30 to 32 ATP. So now thinking about how respiration occurs, there's anaerobic and aerobic. Anaerobic means not oxygen. So it is possible to do some cellular respiration without oxygen. In this case, there is a different electron acceptor that's not oxygen because it's not around. So it could be sulfate, for example, or nitrate or sulfur. There are specific anaerobic organisms that are called obligate anaerobes that just legitly cannot survive in oxygen. So they have to, remain in environments that don't have oxygen. There are also things called facultative anaerobes, which basically are equal opportunity. When oxygen is present, that is what they prefer. It is aerobic respiration at that point because you are using oxygen. It's way more efficient. It makes way more ATP. But if oxygen is not available, it's able to switch over to fermentation, which is the process of doing essentially anaerobic respiration without oxygen. It's not good in the sense of producing a ton of ATP, but a tiny bit of 2 is better than none. So our muscle cells are an excellent example of this. We prefer our oxygen, but then under exhaustion or complete use of oxygen in the muscle cells, they switch over to fermentation. So here's kind of roughly what that looks like. Once pyruvate is made, it's kind of the decision of if oxygen is present, then it will move to the mitochondria, and if not, then it will produce a couple different things. Depending on the organism for fermentation, it will either create ethanol with carbon dioxide, so a lot of like yeast and brewing and baking and things like that, or it can also produce lactate, like our muscle cells. And you get a net yield of 2 ATP. Whereas in respiration, you do need oxygen, but you can generate a lot more ATP as a result. So here are the two different types of fermentation that occurred. As I mentioned on the left, one involves ethanol and carbon dioxide production. The other involves lactate production. So overall, carbohydrates, fats, and proteins are all used for cellular respiration. When we talk about it, we always talk about it in the sense of glucose breakdown, but there's kind of different ways in which it occurs. So that's the end of Unit 3. You have made it past the energy section of AP Biology.